Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. Compared to other branches of computational biology, computational neuroscience has a quite solid foundation. Most of us agree that the neuron is the key computational unit in the brain, and thanks to pioneering work of scientists such as Hodgkin, Huxley and Rall, we now have a well-founded biophysics-based mathematical scheme for calculating how neurons integrate signals from other neurons and generate action potentials. Thus, a course in the biophysics of a neuron is commonly given in neuroscience programs across the world. Today's guest, Christof Koch, has written one of the standard and most thorough textbooks on the subject. His book, Biophysics of Computation, with the subtitle Information Processing in Single Neurons, was published almost 25 years ago and is as relevant now as it was then. Christof Koch has since the 1980s been a leading figure in computational neuroscience, working out of MIT in Boston, Caltech in Pasadena, and for the last decade or so at the Allen Institute in Seattle. In his book, he not only outlines how to model a neuron in biophysical terms, the book also explores how the different biophysical processes in the neuron can do different kinds of computations, in particular Boolean operations such as and or, that are used in making silicon-based computers. Christof has also been a pioneer in making consciousness studies a respectable field of scientific inquiry, and he has written several books on that subject and is in particular interested in the so-called integrated information theory for consciousness, which also has a computational component. Towards the end of the podcast, we also discuss this topic. As for all episodes, information about the episode and relevant links can be found at the webpage theoreticalneuroscience.no. And if you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example, at the cost of $1 per month. As a member, you will also get the video version of the recording with a guest, in this case Christof, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I have also made a YouTube channel called Theoretical Neuroscience Podcast, where you can watch the videos as well. I will be most grateful if you sign up to help me fund the production of the podcast. But I also appreciate all the other listeners and viewers, and I hope that all of you can help spreading the word about the podcast to other people in the community, on social media or otherwise. Note that due to a technical blunder by me during the recording, the sound quality of my voice is quite poor for the first 7 minutes of the podcast. But after these 7 minutes, it becomes much better. But enough talk from me. Here is Christof. Theoretical Neuroscience Podcast, Christof Koch.
Good. Thank you very much, Gaute, for having me on your podcast.
Yeah, perfect. So, just a little bit introduction. I mean, you're a physicist turned neurobiologist, maybe, also with a philosophy training. Is that correct? I mean, you used to be at Caltech for a long time.
Yeah, so my background is physics, was an undergrad in biology, but then I for my PhD, I modeled single neurons. This was back in the 20th century, modeling individual neurons, modeling the biophysics of neurons. And then after a stint as a postdoc at MIT, I came to Caltech, where I stayed for more than 25 years as a professor in biology and in engineering.
Would you say still that you're doing physics? I mean, like a brain physicist, neurophysicist? Do you think it's...
Yeah, I mean, I think my interests pertain to ultimately understanding the universe, everything in it, including consciousness, using physics or physical principles. Yes.
So, I mean, I would say you are one of the pioneers of computational neuroscience. I remember when I started entering the field that was like in the mid 1990s. There weren't even sort of like almost like, I guess, the first book, like was this book by Johnston and Wu that came out, but that was like the only book sort of covering the field. There had been some earlier books on like the cable theory, I guess, with Jack
Noble and Sien, yes,
yeah, which is an excellent book. But still, this was like early years. And if you look back at your production, like your papers. I mean, you have like enormous number of computational neuroscience papers that you published in this, like in 80s and 90s, and also later, right? So, for one thing, they were over a widespread. I mean, they were like a wide spread of topics. So, so is it sort of because it can you say that was sort of early days? So there are many low hanging fruits around, or you, of course, I know that you're very energetic, so you had a lot of work capacity.
Yeah, there are just so many interesting questions to address at a variety of scales, from the very small, the scale of micrometers, particular dendritic spines, all the way to trying to understand local field potentials and EEG and behavior, right, which operates at larger scale in a variety of different creatures.
Yeah. But these are in some sense like quite different problems in some sense. So maybe like in a more mature field, people will have to specialize more, only focus on spines or only focus on local field potentials or focusing more
Yeah, I
on one
think it's
specific.
characteristic for an early phase of a field when you don't have too many, when the field hasn't really differentiated. Look, this started out, several thousand years ago when there was just philosophy, there was metaphysics, and then slowly out of that came science, natural, what was called natural philosophy, which then turned into science, primarily physics, chemistry, and then biology, and then slowly emergence of psychology out of that in the 19th century. And so now we have this this very funny story I can tell you about being a computational neuroscientist. So when I finished my PhD in 1982, I was in Tübingen at the Max Planck Institute for Biological Cybernetics, and my advisor Tommy Poggio had just moved to MIT, where he still is. And he sent me a telegram. Remember that ancient technology, a telegram?
Exactly.
Well, he congratulated me, but he said, well, I'm also a little bit worried because there is already one other theorist in the United States of America, Will Rall, with his electrotonus. And he was worried professionally. He said, now you have a child, you have family, you're married. So you have to think a little bit carefully how you're going to compete with one other theoretician.
That's sort of interesting. It reminds a little bit of, I guess, the IBM boss in the
Yes.
during the war only thought there would be no only room for 5 computers or something,
Yes.
right? So
Yeah,
this
now
was
every
also
university has computational neuroscientists. So the field has become much richer, much more diversified. There are vastly more, people doing theory. Although
but
it's by and large, we will probably touch on it later. It's different sort of theory and computations that people are doing.
because then you wrote this book, Biophysics of Computation, which was published, I guess, 1999, 24 years ago, which I mean, it's certainly a classic. I think it's still the book which has sort of like going through I mean, the biophysics of single neurons in a more detailed and comprehensive way. So, so I certainly recommend people to take a look at that and investigate it if you're interested in this topic. So, and I guess what was interesting, I think, was, I mean, it's not only a book about the biophysics of neurons. It's really about the biophysics of computations, meaning the computations that this neuronal physics might sort of like, well, might support sort of. And I guess that comes from your like, I mean, you came into that via Poggio, right, at MIT, because you had there this program of trying to understand the biophysics of computation.
Yeah, I did. What was unusual, or what still is unusual, I followed my PhD advisor, Tommy Poggio, to do my postdoc with him. So I followed him from the Max Planck in Tübingen, Germany, to MIT. Yeah, specifically, I was in that title. I was inspired by a book that Carver Mead wrote, quite famous one. Carver Mead, who initiated the VLSI resolution together with Conway, he wrote a book called The Physics of Computation, or he had a chapter in a book called The Physics of Computation. In this case, how the physics of semiconductors, the band gap, etc., leads to a particular type of, and not computations in CMOS circuits that are still at the heart of all of our machinery, all of our computing machinery. And so here the question is, what are the basic biophysical mechanisms that underlie computation in the nervous system. Now, a different point of view was advocated for by Francis Crick. So after I started becoming an assistant professor at Caltech, I started interacting routinely with Francis Crick, who at the time had moved he sort of formally retired from the MRC in London, in Oxford, and moved, in Cambridge, and moved to La Jolla, and where he developed an interest in neurobiology, particularly in dendritic spines, and then later on, of course, in consciousness. And he always cautioned me, he said, well, one way to think about these biophysical operations is as computation, but that is not the only way to think about them. And it's true, if you think about what happens in your kidney, your kidney is another biological organ, or your heart, right? You can say the biophysics of a heartbeat, right? And people have done that, Nobel, for instance. Or you can think about the biophysics of kidney cells or liver cells, right? They all have some biophysical operations that underlie them. But no one talks about the computations that your liver does, or your computation that your kidney does. And why is that?
So we come back to this computation, but first I'd like to focus on the biophysical modeling of neurons, right? Because what really, without thinking about necessarily what computations the neurons can do, because what sort of, if you sort of look at the book chapters, right? And it's like about the cable equation and ion channels, Hodgkin, Huxley modeling, and so on, and all the synapses, and it's not that much that has happened in the last 25 years. Or 24 years since the publishing of the book.
Well, I mean, there's been a vast proliferation of much more detailed models. But yes, the basics of that was established by cable theory and by Hodgkin and Huxley, in their 4 papers in 1953. That's true.
And Rall, of course, with the dendrites.
And then all the electrotonus calculus for dendrites, and everything since then has been an elaboration. Well, I mean, there's this entire ionic channels, right? None of those theories accounted for, the discovery of binary, discrete, stochastic ionic channels.
And we made that based on patch electrodes?
Yeah, by Sackman and Neher.
Yeah.
So underlying, so we have all these currents, Hodgkin, Huxley, and all these are macroscopic, deterministic, continuous currents. Underlying them, we now know, are discrete, microscopic, all or none stochastic ionic channels, right, or other channels. In all the modeling, the vast majority of modeling, 99.9%, that level is neglected in favor of this continuous, deterministic, macroscopic level of currents.
One thing that also has happened now is that you can do these molecular dynamic simulations of ion flow through this ion channels using amino acids representation and so on. But that's also not something which is extremely computationally heavy, and it's not something which is sort of typically used, right?
That's correct. It's not used in neuroscience. I mean, people are doing it for doing drug discovery and drug design, right? Trying to understand from a bottom up using simple description of individual molecules and their, Coulomb attraction between them. But it's not part of neuroscience. In fact, neuroscience has moved the opposite way, right? The vast majority of neuroscience model abstract even away from Hodgkin Huxley. More and more these days, they even abstract away from the reality of spiking cells in favor of large number of continuous neurons, partly inspired by the incredible, amazing success of, deep machine learning and their representation of, quote, neurons.
Yeah, because so you think about what's called neural field modeling and neural mass modeling, or of course in the basic unit in the AI is this, I guess, ReLU, or this this threshold unit, which is also based on continuous.
Well, either it goes back to McCulloch and Pitts, right? 1943. Or it goes back to models, Fukushima and other types of models that are continuous, very early days, and then Hopfield. And so people use those models, but then, of course, in networks that have 10, 20, 30 layers, where usually they look at firing rates, continuous firing rates, and continuous input and outputs.
Mm. Yeah. So, because but some things which has, I guess, spike. When we think of synaptic dynamics, I guess the basic sort of models, at least phenomenological models for the synaptic currents, these conductances, which is the AMPA and then the A and so on, they were already known essentially when you wrote the book. But the spike timing dependent plasticity that I guess it was discovered about the same time.
Yeah, so that was already around, that was the 90s. Yeah, the basic biophysics of synapses is, Katz and Eccles and people like that, right? So all of that was discovered in the golden age of biophysics, really the 40s and 50s and early 60s. And so we've learned vastly more details, but the principles haven't really changed that much.
Interesting, yeah. And then you have, I guess, also the dynamics of extracellular space. That is something which has maybe
that
developed.
That is pretty recent, yes. Yeah. And the realization that may be critical, that in fact one of the functions of sleep may be, relate to that extracellular space and extrusion of various substances in that extracellular space.
So that the volume change may be actually used for something and
used for something or has to be taken into consideration. And in fact, that this may even relate to pathologies like Alzheimer's, right, where the clearing in the extracellular space is insufficient. So that's all pretty new.
Yeah. What about homeostasis? This idea. There's something about like a neuron. If you have a neuron that is a burster, I'm finding like it's burst spiking. Then if something bad happens to one of the ion channels or something, it typically comes back to being a burster. So there's some kind of homeostatic rules that keeps the neuronal identity, right? So these are maybe, I guess it was some work earlier.
There was work done on this, on development. How do you get actually to a Hodgkin-Huxley neuron, right? So let's just take the classical Hodgkin-Huxley, right? How do you, every neuron early on in as a fetus, right? So early on neurons don't spike. Around the second trimester, neurons start generating Hodgkin-Huxley-like dynamics, and you can also see that in the EEG. The EEG in preterm infants, very early preterm infants, start showing these burst and pulse patterns. So you have to think: how does a neuron wire itself up? How do you develop the exact, given that you have these components, the sodium-potassium channels, how do you adjust for the exact balance, right? And there have to be learning rules that need to be involved in that, let alone the much more complicated things like the bursting you mentioned. There's surprisingly little work done on that.
Yeah. Because that could, if you, in terms of like fitting model parameters, say if you want to make a model neuron that represents or many type of different synaptic inputs, say, then often you fit these conductances, like the values of these conductances. And I guess if instead we had models at the level of sort of essentially learning these conductances, or transcribing and making these proteins that makes up what I'm trying to say. You could have these learning rules that sort of makes this conductances regulate itself, so that in some sense what you have to find in the model are these learning rules.
It has to be something like that. It has to be a self-supervised learning process around, let's say, a calcium set point hypothesis, right? The idea is that on average there's going to be 50 nanomole of intercellular calcium. And now adjust slowly, adjust your conductances, your sodium, potassium chloride, whatever, to achieve that. It has to be something like that, because
and I think that was actually, I think it was done earlier. Neve Marty's group with David and Larry Abbott. And,
yes, exactly. So people have looked at that.
And also with Tim O'Leary,
not that much work, yes.
So that would be an interesting thing. But what we don't know anything about, this is typically about sort of mature cells and actually like static cells, more or less, even though it has plasticity a little bit. But development is still not so much known. Very little computational neuroscience, and there are, but there's like not on there's more phenomenological modeling, it seems.
Yeah, I don't really know any detailed biophysics cellular models these days that pursues, how do you go from a stem cell to a precursor neuron to a spiking neuron, actually modeling that transition, right? Which has to involve a combination of cell biology and cellular biophysics. I don't know anyone who does that.
Yeah.
But, the field is so large. There are, 50,000, 60,000 neuroscientists.
Yeah, but
these days.
how many computational neuroscientists there is? Like maybe 10%?
Yeah, but that's my gut feeling, probably less than 10%. But most of them are concentrated on learning in adult tissue, as you mentioned.
Yeah. Okay, so now we talked about the biophysics of the neurons in itself, and now the biophysics of the single neuron computation, which is sort of the main topic of our book. So what is computation?
Well, the way I think about it is it's a mapping. It's a high-level mapping from the physics of your mechanism you're investigating, whether that be CMOS and NOT circuits or neurons, onto a mathematical calculus. In particular, mathematical calculus that you believe underlies some sort of higher order abstract operation, like extracting variables or computing invariances, right?
Not not heartbeat, but
thinking. Yeah. So why don't people think heartbeat is a computation, or why don't we think gravitational forces acting inside a black hole are computation? Because we do know the brain, particularly the human brain, is capable of doing high-level logical inferences, right? And ever since there's tradition going way back to Leibniz, I would say, right? And his logical calculus, when he said, well, instead of having an argument, we should just resolve any argument by saying, let's calculate, calculamus, right? And then through Gödel and all of that, to the modern McCulloch-Pitts, etc., the belief that we have this capability, the capability to perform these abstract computation, and that's also operates at lower levels, including the eyes, including all the sensory and the motor systems. And it's the one overwhelming paradigm we all grew up with, and we are suffused. The world is suffused with it because of computers, because the enormous success of computers.
And you're referring to this McCulloch-Pitts, and that was his 1943 paper, where I guess the idea was to make actually some kind of Boolean logic based on these neural units, right? So
exactly, to show how biophysics or simple abstract models of neurons is capable of carrying out any logical operation, right? Any first order logical operation together with memory, right? They weren't really concerned with memory, can be instantiated with such neural networks. That's really almost like genesis of the field of computational neuroscience. That's what I would say. That's when it happened. Because here it was explicitly they used the word and computation. While Hodgkin and Huxley, although it's critical, they were more just interested in the biophysics of action potential propagation. So there was less about computation and more purely about biophysics. So I think you have these two origins. You have the physics or the biophysics of operations in the nervous system, going back all the way to Helmholtz, another physicist. And then on the other hand, you have this computational side. Starting with Hodgkin, with McCulloch-Pitts, and then, of course, with the perceptron, and then, Hinton-Sinofsky, the neural network revolution, deep machine learning, and now generative AI.
Because if you look at the computations done in computers, there the main signal carrier at the physical level is, I guess, it's a voltage across this
charge. It's electrical charge shuttling across, from one gate to another gate. Yeah, and then that triggers the gate voltage, and that changes, opens or closes the gate, and then you have a current flowing to the next transistor.
And because I guess if you look at the neurons, and there are two sort of obvious candidates. One is the membrane potential, which of course reflects charge, and calcium concentrate, or you can have these signaling molecules like calcium, right? Or maybe other things as an information carrier.
In principle, neurotransmitter, your serotonin, your cyclic AMP, right? The brain doesn't make this distinction between what's used for signaling and what's maybe used for metabolism, right? Because it's all sort of one continuum, and it has to watch very careful that the electrolytes don't get out of balance, right? So that's different from engineered system where we carefully engineer to minimize these interactions, right?
So, I mean, okay, so if you then look at sort of the logical operations or computations that neurons potentially can perform, and I guess one thing is the thresholding, right, in terms of spike generation. That's sort of like a nonlinear, like a diodic, I guess corresponding to a diode in electronics, right? So that's one. Again, I guess that's at present it's the only one used in AI. These deep networks only have this thresholding. They have other
Correct.
things. Yeah, they don't
And
have other things.
halfway rectification and thresholding. Well, again, because it turns out, going back again to McCulloch Pitts, that's universal, and anything that you want to compute can be instantiated sufficiently number of them, and we have in Cortex 16 billion, and that seems to be enough for at least that's a hypothesis, seems to be enough to explain most of cognition.
But nevertheless, in your book, you explore all these other possibilities with dendrites, like shunting inhibition, for example. What is shunting inhibition?
Yeah, so shunting inhibition is the fact that if you open certain types of synapses, particularly those associated with GABA A receptors, they open at a membrane potential around rest, so -70. So that doesn't really inject so much a D or hyperpolarizing current, but just shunts. It opens a conductance. So it'll now the same current will lead to a smaller voltage change, right? Just following Ohm's law. And then you can I showed as part of my thesis that shunt can be used to compute very specific things, not only saturating nonlinearity, trivially, of course, but also can be used to instantiate and not gate. So very specific types of logical operation inside the dendritic tree. So here the idea is that
what's an and not gate? That's
well, if you think about it, it only fires when you have and not. So you need 1-1 in order for it to be active, right?
So yeah, exactly. So you need an excitation,
and then not an inhibition.
Yeah, exactly. I guess that's what it's.
Yeah,
that's what it needs. Yeah, and you also yeah.
And in principle, then you can doing that you can again you can compute anything you want to compute.
Yeah.
So the question is, to what extent are these as such and others? You can think of NMDA spikes, you can think of calcium spikes inside the dendrite. To what extent is there a rich computational substrate inside neurons, rather than saying, well, neurons just sum it linearly as they do in all neural networks, and then you have the somatic or axonal threshold.
Another example you mentioned is this: if you have spatially clustered NMDA synapses, you can have this multiplicative mechanisms that you can do some kind of multiplication.
Yeah, using either and not, or using NMDA. So there is in principle a rich substrate of mechanisms. These NMDA synapses, NMDA triggered spikes, calcium spikes, shunting inhibition to perform complex operations inside the dendritic tree. It's difficult. I mean, people are of course studying that, but it's difficult to see that because it's so far away from the soma or the axon hillock where most of the recording is ongoing, in particular in living animals, compared to in slices that are sort of close to a comatose tissue.
I guess, I mean, if you have all these different mechanisms, there are also other you mentioned in your book for doing, which in principle are sufficient to do the necessary computations in some sense, and I guess the real brains, and this is we're now even at the single neuron level, we're not at the network level yet, where of course other things and other mechanisms may also pop up, so they could be at the single neuron level. You could have like one or more of these mechanisms, right? And they could be working in tandem, and are we any closer to? Have you learned in these last 25 years any more of which of those are actually used?
No, I think sort of the field has, correct me, I'm wrong, Gaute, but my feeling is, with the exception of dendritic spikes, that clearly people like, Matthew Blackham, for example, think are very important, including up to performing a critical operation for a sort of handshake that's involved in consciousness. By and large, people don't focus so much on that single, what happens in dendrites anymore. It's more what happens in these collection of very large circuits. But maybe I'm wrong with that. But the sort of, if you look at the majority of papers published in high-profile journals like Nature, Science, Neurons, etc., they tend to be more at the network level rather than spending a lot of time inside dendrites, figuring out the exact type of logic. Partly because of the realization there's so many mechanisms, and the brain isn't concerned, unlike engineered product, the brain isn't concerned so much with necessity and sufficiency, right? Because it's an evolved system. So usually there are many parallel mechanisms, each of which may not be sufficient by itself, that carries out any, there's a huge extent of redundancy and degeneracy. Right? Every evolved system is characterized by redundancy and degeneracy. While we like redundancy in our circuits as well, right? We want our computers to be robust to damage. We don't really think about it a lot. And we certainly don't think about degeneracy in our engineered systems, in our engineered products.
So one thing which is like an intriguing finding maybe is that you have some of these examples of neurons with dendrites that have a bunch of active dendritic conductances, but somehow in the end they end up some kind of conspiracy between these nonlinearities to make the functional behavior quite linear. So that it's sort of just like as linearity is important, at least for this neuron.
Yeah, I'm a little bit more careful because much of this work, again, is done in slices. And, it's very different in a living, breathing, thinking animal. We like to think about linear because, clearly it makes our life much easier. Things are linear. There's no reason whatsoever that, evolution cares about linearity.
No.
Right? In fact, it has to compress signals, things like photons, the rain of photons on our retina, there's 10 orders of magnitude fold variation between moonlight and bright sunlight. So clearly, there have to be all sorts of adapting phenomena.
Exactly. No, that's true. Yeah.
So we like to think about linearity, but evolution doesn't care, and so we look for it, and we are very
But
proud of it when we find it.
linearity is an advantage, some aspect of signal processing. So, but evolution finds it.
Yeah, although for any real decision making, involves some
That's true,
sort of
yeah.
extent of nonlinearity. And as I said, the vast range of physical signals. Just if you look at variables like pressure, pressure, light, etc., chemical concentration is so big that it can't map in any linear way, let's say, on action potential frequency, right? Because that only varies across 1 or 2 orders of magnitude. So there has to be massive compression, but a smart compression and sensing going on. I think it's primarily a bias that people have, because that's the way we think.
I think one really example, a nice example of is sort of the work you did on this, I guess, together with Gabbiani and others on this locust movement detector. So that was, I guess, if you sort of map to this, to this Mars level of description, you have this computational this level, which is sort of what's the function and what you want to achieve, and that's algorithmic, how sort of like algorithms for doing it, and the mechanism. I guess so far we have talked about maybe parts of algorithms, or at least its logical operations, things that can be used in algorithms, and then the mechanism. But for this locust movement detector, there it seemed to be like a one neuron that covers all of this, right?
Yeah, or 2 neurons, that are directly presynaptic to each other in tandem. Yeah, so there you have this nice the way that the locust does it for this emergency warning, this looming when something comes at it at high velocity. so it can jump away, seems to be that you first go into log space, and then you add variables in log space, and then you go back to exponentiate, right? So
because when you add in log space, that's multiplication, right? So
yeah. So that's a clever way that particular critter did it. Yes. Yeah, and there'll be all sorts of interesting shortcuts like that in our brains too. Of course, the locus has its other constraint that we don't have. It has to fly, right? So whatever computation you perform, it has to be performed in a very small volume.
That's true, but it also has to respond fast, right? So
And it has to respond very fast, yes.
Yeah, to get away from the predator.
Yes. I seem to remember it start doing it within 30 milliseconds onset. Yeah, so it's very
That's
fast.
why it's difficult to sort of to catch bugs with your hands, right? Because
yes, correct.
So, but one general question is sort of this. I mean, I just read this book by Bushaki, like The Brain Inside Out, and there he made this point, sort of that that neuroscience has sort of spent too much time trying to map sort of the brain to sort of this man-made notions, like in psychology, attention, perception, and so on. That's maybe trying some sense to fit the landscape to the map, rather than going the other way around. I'm thinking of, in terms of logical operations, this thing of trying to map like neuronal processes to logical operations. Is that also a natural thing to do? Is it sort of a...
That's why Francis Crick always said, my boy, be careful. The way he thought about it, he says these are biophysical operations that lead from, let's say, the range of photons on the retina ultimately to behavior. We can all agree on that. Whether or not it's useful to think of them as computation depends. Now, we, as we've said before, we cannot help but think about computation because we use computers everywhere, right? And so computation is at the basis of everything we do. So it's very difficult to escape that. But why doesn't the liver compute? Or why doesn't a black hole compute? And in some sense, you can say, yeah, any physical system computes, but then, of course, the meaning of computation becomes meaningless. So it's really, and Buzaki is totally right, it's this extrinsic point of view. We look at something like the brain, we try to analyze the signal flow, and then we assign, because we want to understand it, and our ways of understanding involves abstraction and mapping it onto computations. In particular now with computers coming closer and closer to us. To our native intelligence. And so I think this is even more powerful now than ever before. Biology doesn't care about that. Evolution doesn't care about that. It just cares about making sure that you're around long enough so you can pass on your genes to the next generation.
So, I mean, if you sort of look
at particular, by the way, we'll probably come back to it later. This external versus this intrinsic. You can also call it the extrinsic point of view versus intrinsic. Particular matters once you start thinking about high-level things like meaning. Because meaning has to arise intrinsic from the brain. It can't be imposed from the outside. And if you go inside the brain, it's pretty dark inside there. There is, there's no light inside the brain. There's no meaning. So how does meaning get into the brain?
So I guess that happens. The networks are involved. And I guess one thing that has happened really since you wrote this book is that there's now much more network simulations. I mean, of course, there were sort of like examples of this also from, I guess, early 90s. You were involved in like some early visual, like some network modeling, right?
Of visual cortex, of retina on Lisp machines at the AI, when I was at the AI lab at MIT, yeah.
And then you had, I guess, one sort of like seminal paper or like a milestone paper was the paper of Roger Traub, on the making this model for the somatosensory system with this like 3,500 neurons, and like even though they were sort of like some kind of stylized neurons, they were sort of quite complicated. And then that has moved on, and I mean you have the Blue Brain project, and also with the large scale network of neocortical column, and I guess that's also has now been expanded in a recent paper that came out this year. And then you have also you at the Allen Institute have also made this large scale model, well, together with Anton Arkhipov and you on the mouse V1. So, and you had actually a quite nice sort of like a topical review just out on this in a journal of neurophysiology,
Yes.
which I sort of recommend. So, but first, what is sort of meant by this sort of? I mean, these models are what's called biophysically detailed neural network models. What is meant by that?
Well, ultimately, it's all very much in the eye of the beholder, right? Because clearly there are no ionic channel in these models, and we know ionic channels underlie everything.
Yeah.
So, some
So what do you mean
people,
that there are ionic channels in your neurons?
no, there are none. It's all continuous current. It's continuous Hodgkin-Huxley like
Yeah.
current, but
Okay.
in the
Okay.
model.
So the ion channels are represented with these Hodgkin-Huxley
Yeah.
models. Yeah.
So I mean, when people say, when I say or you say biophysical accurate, it
Uh,
means we have some biases. In this case, we think it's important to
I don't
represent.
say accurate. I say detailed. So that's, but
Okay,
still, there's
detailed.
limit. Yeah, it's still limits
Okay.
of detail.
Yeah.
Yeah.
You still have to make a selection. What you think are the relevant details, right?
Exactly.
So people like you and I, we believe what's relevant. A is the dendritic morphology, right? Clearly, it's important to model because neurons differ dramatically in terms of their extent and the geometry of the dendritic tree. All right. And that's important because there are nonlinear interactions within the dendritic tree, right? If it's all linear and they have the same weight, then you don't need to model it. And so then what else is important? Probably active conductances, dendritic spikes are important. What else is important? Well, probably the dynamics of spikes, because they ultimately, and the dynamics of synapses, because both of them together give rise to these large scale movement of charges that ultimately expresses itself in changing extracellular fields and in things like the local field potential that you measure outside with your electrode, and or ultimately, of course, even the EG that you can measure from outside the skull. And those things you don't get if you neglect much of biophysics, right? If you have point neurons, you don't get it. If you have just Hopfield neurons, you don't get it, or McCulloch-Pitts neurons, you don't get any of that. So if you care about those things, then you need to go to what's called detailed biophysical models. But many people don't care, right? If you just care about, how does the brain carry out, how does my model carry out some invariance computation, you may not care, and you're just as well off with leaky integrated fire neurons.
But say if you are ambitious, then or ambitious, or if you want to be able to have models that compute not only spikes and membrane potentials, but also then like these local field potentials and EEG and what have you, and you want to mimic a particular, make a model for a particular system, like the mouse primary visual cortex, for example. So what? How then do you what? How do you go around sort of or go about developing or how do you what is the goal of that model? What should it explain? How do you evaluate whether it's a good or bad model?
Yeah, these are very good questions. A, how do you select your details? So we probably know more about visual cortex now than any other piece of cortex, just because so much effort has been expended on it. Do you model all the different cell types? There are roughly 100 different transcriptional identified cell types. Do you include all of them or just a subset? And then do you include all the vast, diversity of different ionic channels? You have to make some sub-selection. And then the question you asked, how do you test this? So you can clearly only test it in some statistical, meaningful manner, right? Where you show, for example, physiological input. I think it's critical to test it to approximate what happens in living animals rather than in slice, because slice is very far away, right? Slice is a comatose brain. That's fine for studying individual neurons, but if you want to study networks, you got to study it in the living system. And so then typically you say, okay, here I have some collection of stimuli that I show to the animal, and this is the response using local field and spikes, and I try to replicate that. Even that, of course, already involves statistics. You have the average of multiple trials because there is a lot of trial-to-trial variability, which ultimately you also need to explain. So it's challenging, and the question
You
ultimately,
this little, yeah, this you call it the biological imitation game. I remember in the paper you wrote.
Yeah, so one way to do it, so I introduced together with Michael Bice, sort of a la Turing, the imitation game. So here, behind on the one hand, I have a real biological network. On the other hand, I have my 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 model that's supposed to model it. Can you tell where the difference is? And how would you tell that? Well, one way, of course, you can always ask more detailed biophysical question. You can always ask, well, for example, you see evidence for all non-ionic channels, or you don't. So then it's a model. I mean, it really begs the question, Gaute, how do you ultimately, what is even a correct model, right? If we just stick with V1, what does it mean to say I have a correct model of V1? Because we know, let's say, talk about the mouse to make it a little bit more bounded, right? Let's say adult mouse. Let's forget about development, right? Because one mouse, if you do electron microscopy, EM reconstruction, each mouse is slightly different, right? We know this. And then, as I said, there's this huge trial-to-trial variability, right? That's why you have to do the same thing 10, 50, 100 times and average over that. So what are you trying to capture? It's not an easy question to ask.
So kind of average mouse, or is it an individual mouse, for example? That's one question.
Yeah, I think ultimately it has to be some sort of statistical average that I capture 90% of the variability of what a mouse experiences in a day, let's say, or an hour, some suitable time span, or over a few minutes.
But I also think, yeah.
Anything beyond that means, I want to answer what happens in this specific mouse.
But it's also this, I mean, if you sort of, I'm thinking of when you look at sort of, when you fit sort of like a deep network, a convolutional neural network. And if you do that to, say, if you have a set of like images that you want to learn to categorize and so on, if you do the training in like different orders of the images, you will end up maybe with two networks which are equally good in terms of functioning, but their details, like synaptic connections between the layers, will be different, right? So that's all.
So this is what I call degeneracy, right? There's both redundancy and degeneracy, right? So and any one operation can be done in myriads of different ways, particular biology. And then there's the redundancy. You can knock out individual mechanism, and the system will often continue to do its job more or less intact.
So yes, I remember I was I remember I was actually, when I started just entered neuroscience or computational neuroscience, I was at a meeting in Sweden with where this John Hopfield was there, and I remember he started his talk saying, in computational neuroscience you have to learn to compute without knowing all the numbers, and that's very difficult. That's sort of what you have to learn to do in some sense, right? Because we are so used to, when we have a model or a physical system, we fit the model to data, and maybe the model has so few parameters that it has a unique solution, and then that will sort of declare victory if you find that unique solution. But that's a very different thing for biological system, particular networks, but also single neurons. It's the same. We could always parameterize a single neuron model so that it's sort of like it's sort of lots of degeneracies of the many solutions, right?
Yeah, and I think it's also going to be true inside. Once you try to understand, even a small cell contains on the order of 5, 6, 7,000 different proteins that are all in, many of which are in solution. Maybe a billion molecules, and there's huge degeneracy and redundancy there as well. Yeah, I think that's the character of any evolved system.
But the same, if you were able then to develop this, say, a mouse V1 model that along the lines that you are working on at Allen, and which are then able to predict essentially all the responses to, or at the responses, it's sort of that the responses in from the model is indistinguishable from the responses that you can measure with an electrode, like your NeuroPixel Pro, on a real mouse. And if that worked for all stimuli and maybe different brain states and so on, that would be like the ultimate, or that would be really cool.
Yeah, so there would be progress, particularly if you could do it in different brain states, like particular sleeping, anesthesia, non-REM and REM states, and behavior. No one has done that at this level of biophysical detail. But of course, we all know, V1 is just, it's a tiny, grain of quinoa size piece of cortex. It doesn't hang there in isolation. It's attached to a lot of other brain matter. So ultimately, we need that for the entire brain.
Sure.
And the question is, will we get there? In terms of raw computational power, yes. But then, as you said, will it be a model, let's say a deep, and people have already in the visual system of the monkey, let's say, right? Jim DiCarlo and his group at MIT, right? As you mentioned, they have these models that can reproduce a spiking response of neurons higher up in the visual system, right?
Yeah.
Without being biophysical accurate. And maybe that's a relevant level. It really depends on the question you're trying to ask.
And I guess, one thing, if we're able to make such a model, say for the V1 or even the whole visual cortex or even the whole cortex, one thing it would sort of be, it would differ from previous net, because if you look at the visual system, there are all these models that has been developed over the last decades, which explains individual things like contrast invariance or directional cell activity or orientation cell activity. But the hope is that this, which are in some sense single purpose models made to predict the one particular thing. But such a model, if it works, would in principle be multi-purpose, right? Similar to...
Well, I would hope it would predict existence of Jennifer Aniston neurons, for instance, right? But it also has to predict what happens as the system closes its eyes and goes into a state where it maybe dreams of Jennifer Aniston, right?
Well, that's
we do this every night, right? Maybe not with Jennifer Aniston, but with other people relevant to our lives. And so yes, we're still a ways away from that.
That's ways away. But I guess it's also then you could say, well, then the simulation becomes almost as complicated as the real brain. So you don't really understand it, but it would be a perfect starting point for getting more these qualitative, like simpler IDs and simpler models. And so it would be like, I guess you need a set of models at different levels of granularity and complexity to think that you understand it.
Well, it relates. What do you mean by understanding?
Yeah, no, that's of course true. But I think...
What do you mean? When you say understand the brain, what do you mean?
Okay, so maybe I can make... I used to do semiconductor physics, so condensed matter physics, before I came into neuroscience. And there, in some sense, you knew that if you're able to solve the Schrödinger equation, this many electron Schrödinger equation for your piece of material, metal, a semiconductor, you know that everything is in there, like superconductivity and all these quantum Hall effects, all these fancy effects. So in some sense, if you're able to get it through this large scale, like simulation with solving the Schrödinger equation for all these electrons, like brute force, that would be, then you would know that, okay, that is at least sufficient to sort of account for this phenomenon. My model is able to predict sort of like this outcome. And then if in addition you had like these simplified models like you have for the superconductivity, what's called this BCS, Bardian-Cooper-Schrieffer theory, where you assume something, that some term of this Hamiltonian, as they say, is particularly important, then you can sort of understand it more like with a simpler model. It's like so I think this combination of models at different levels or with different concepts together maybe form an understanding. That's this for me.
That's true for physical systems that we have studied in the past. It's less clear whether that's true for a complicated physical system like the atmosphere. Right? What I see now in atmospheric physics is these gigantic weather simulations that are now, by the way, coupled more and more with generative AI, right? Right, where they use machine learning to replace rather than doing forward physics computation, you lose learning, where you say, well, this pattern will 2 weeks later lead to that pattern of weather. But biology is just vastly more complex, and I think you can have. As we mentioned before, my wife just was infected by COVID and took Paxlovid. And Paxlovid is a beautiful instance of understanding viral replication at the mechanistic level, at the level of molecules and which protein are involved in leading to replication, and how to interfere with certain types of proteinases so you can prevent replication. And it works incredibly efficient. So it's really a powerful example of mechanistic understanding. By and large, we don't have that in neuroscience. We certainly don't have it comprehensively from, let's say, the level of ionic channel all the way up to the level of
But the model you talk about, the mouse V1 model, that in principle could have that in the sense that it sort of has receptors in there that the medicine molecules can sort of interact with. So it's we are not there yet, but in principle, it's at the level of.
Well, in principle, yes, it's a building block. But look,
Yeah.
this model doesn't predict why. I know this. You show the animal 10 stimulus, and twice it may not respond at all, the same piece of V1. And 3 times it responds with, an up wave. It may be in a down state. It may whisk. Then we know, even though, the animal is witching or whisking with its, with its whiskers, that influences V1. That's not in the model, right? The fact that this model, that the real V1 can go to sleep, right, or can dream, none of that is in that model. And I'm not trying to poo-poo this model. It's just, it's one of its progress, no question about it. But it's a long way to go to really have a model that captures all the vast variability. The fact, for instance, that almost no piece of the brain is isolated. No piece of brain is isolated from any other piece, and we now see these with these neural pixel recording. Right, you can see where spikes of, for example, for learning, for reward, for you can see the influence of spikes throughout the entire throughout the entire brain, including the visual system. So it's very difficult to isolate, to say, well, this is just V1 and all it gets is just V1 input. In the real animal, V1 is influenced by many, many other factors, such as reward, such as hunger, such as motivation, such as attention, arousal, pupil diameter, etc.
And I guess there are like two conceptually different problems. One is that it's the model of the connection to the V1 is not well enough model as it is. And then the second thing is that we don't know all the other input to the V1. So it's like that's a two different kind of.
And we don't understand the modulation. There are all these neuromodulators, famously serotonin, right? Oxytocin, acetylcholine, etc., all of which we know the receptors are distributed throughout, noradrenaline throughout the brain, including in primary visual cortex. And we know, depending on our various states of arousal and other things, they can change the property of individual neurons. We know this a little bit, but we don't understand enough about it at the quantitative level to be able to model it.
But if we go back to the weather, just this question, what does it mean to understand something? When, if you look at the weather, I think we all know, well, we know the physical processes like with pressure and temperature and humidity and all these things, we know pretty well. And we're able then to put it on the computer based on these equations and compute the weather quite accurately into the future. And even though we sort of we don't, well, and then we can sort of put in terms like sort of like tornadoes or whatever, fronts and heat fronts and whatever. But even though we don't have like many intermediate notions, like the way to describe it between sort of water droplets and hurricane, still it's, if you're able to compute it and predict it, that's one kind of understanding, right?
Correct. I agree.
Yeah.
If we can predict really things reliable, but then of course, how far out? Is it 1 day? Is it 2 days? Is it 3 days? And at what level of granularity? Can it predict rain? At the level of a kilometer or at the level of 100 meters? Yeah, but predicting things is one way to say I understand them. Although the classical understanding is really more informed, by things like quantum mechanics or relativity, where we really understand, there's a simple set of laws, let's say 10 equations, and you can directly from those equations, without invoking, huge supercomputers, predict, let's say, the shift of the orbit of Mercury, right? The shift in its perihelion, right? That's sort of the classical model of understanding, or the structure of the chemical elements in, why we have 92 elements, right? You can sort of still understand that at the level of an undergraduate. But for example, we now know there are 5,000 cell types. There's in human or the mouse, there are 5,000 different types of cells in the brain based on transcriptional criteria. I mean, who's going to understand that? Even if that, assuming that fact is true, who's going to understand that? Who's going to make sense of that except some, super duper computer model?
Yeah. In your last paper, I mean, this review, what's called a perspective review, whatever you call it. So you also mentioned this. There's some others compared with some other sciences where they do these large scale computations. And one thing you pointed to were these cosmological computations. They are really cool because then they simulate the development of all galaxies. And I guess the number of galaxies are even in the universe is even larger than the number of nerve cells in the human brain. So there's
but their laws they essentially scale with distance, right? What galaxies don't have, what we have, are WIJs, right? So I can have synaptic
WIJs. What do you mean by that? Yeah.
Well, I can have synaptic weights that fall off completely nonlinear with distance, right? Right? It can be very specific with some neurons over here and over there, but not all the intervening neurons. This doesn't happen. The gravitational interaction scales with 1 over r over long distances, right? So you don't have this specific point-to-point interactions among stars that you have among neurons. This is why I think the brain is by far the most complex organ of active matter in the known universe, because of these incredible complex nonlinear point-to-point interactions.
That's true. On the other hand, we know that it's some kind of structure in there because it works. Well, like a collection of galaxies don't work, or a waterfall don't. That can be just noise in some sense, right?
Well, in one case, I would say they've been operated on by natural selection. As far as we can tell, there's no natural selection operating at the galactic or even at the planetary, right? There's no competition for scarce resources, right?
No.
So in that sense, no, they're simple formed by purely physical laws. Biology, of course, is subject to the same physical laws as galaxies, but you have competition. And competition gives rise to all these, very far from equilibrium operating, highly nonlinear systems, us among other.
You mentioned a bit this use of AI. So do you know any much? There's this famous AlphaFold that was able to predict all this, like I guess ion channel structures, like protein structures. I guess protein
structures from their sequences, sequence data. Yeah.
Are there any sort of? You at Allen, you're dealing with enormous amounts of data and a wide variety of data. I guess so. Like now with all this EM data that is coming out from the mouse, which is very exciting, but also quite difficult to analyze. Do you use AI in?
We use a variety of learning algorithms. And I know there are people building ChatGPT for biology, right? Clearly, proteins are, and we know enough about proteins, and you can argue that the structure of language and the structure of proteins and protein folding has a lot of similarity, that we may be able to use large language models, or that we may be even able to use similar structures to large language models to better understand, these very complex molecules of life. I know it's a very active area of research, and I think it's going to happen for neuroscience too. Ultimately, that you won't need physics, that you don't use sort of, a simple equation, to evolve a system using physics or approximation of physical system, but that use deep AI or generative AI to predict what your network will do. And maybe this is going to work much better because you can bypass a lot of the tedious if you have enough training material. So we do have that for cells. We do have that for we should have that for EM data. We do have that for protein data, but whether we'll have it for neuroscience, for systems neuroscience, remains an open question.
I guess you already have like for example, some work from Andreas Toller's lab on making these foundational models where they're able to train convolutional neural networks to predict essentially like responses to two photon imaging responses to different kind of. So that seems to be really efficient and really effective in.
Yep, and I think the same thing is going to happen for biophysical, ultimately for biophysical models, and maybe that's a better way to deal with this vast complexity. It's not the type of computation that I was raised up and I learned, but
Nobody
that's fine.
was raised with this.
No,
at least with our age.
No.
Yeah.
And so maybe that's what it ultimately means to understand biology. I can have a generative model that is indistinguishable. If I can, because following the imitation game, right? If I can tell this generative model from the real thing, well, then, it's good enough. It's good enough to fool me. That's good enough to achieve some sort of understanding. Of course, it still leaves open the question, can I then predict things? Because these are not causal models, right? Can I then predict completely new classes of phenomena? Can I take this model of Andreas Toller's and, add a drug like, for example, psilocybin that we know goes here, magic mushroom, that goes to 2A receptors. And of course, the system won't respond to it because it's never encountered a single example.
So I
think all of this works well. These generative models work well when we really understand the MO, the mode of operation of these systems, and we can explore a large fraction of their typical states.
So the training data sometimes includes all the situations you're interested in.
Yes. Or not all, but a large fraction of them.
Exactly.
That's sort of, you cover your training space with samples from each. If you have totally new phenomena, like dreaming, his model doesn't replicate what happens in the visual system when the animal dreams, or on a psilocybin, or on Alzheimer, or many other conditions.
I guess that's the thing with this. If you are able to make these mechanistic models at the level of, well, say, biophysically detailed neurons, if that is sort of like sufficient, then in principle, then you could do this test with drugs and all. And if it was perfectly, then in principle, it should be able to compute it. Of course, it's a long way from now, but.
Yeah, but it also means in principle, then you have to test it, right, on 2A receptor distribution, right? And what happens in REM sleep versus non-REM sleep. And we all know that's very difficult. So it's going to be hard either way. But then we want to keep our job.
Yeah,
if it would be easy, someone would have done it already.
So finally, of course, now the last, I guess as a side project, main project, you've also been very interested in the question of consciousness since you met Francis Crick and you worked with him and you were sort of like one of the founders of sort of the making this into like the scientific quest for trying to understand consciousness. And you've written a bunch of books. And I guess one was the Quest of Consciousness, 2005, Consciousness, Confession of a Romantic Reductionist, 2012, The Feeling of Life Itself, 2020, and now there's a new one coming out, isn't it?
Then I Am Myself, the World.
Ah, excellent. So when will that be out?
Next year.
Next year, 24?
Yeah.
That's good. Because most of these theories don't really have like computational aspects, but I guess your favorite theory, the integrated information theory, does, right? Or you have to have this.
Yes and no.
Yeah. Okay. Tell about the theory first, a little bit.
Okay, so one of the more popular theories of consciousness is this integrated information theory. It says that ultimately consciousness is not a computation. It's not a process. It's not a message that's being sent. It's a structure, and it starts out with making some assertions about what exists. So in some sense, it's a very fundamental theory because it starts out with existence itself. What do we mean that something exists? The theory says something exists. It must have causal power. Same principle in physics, although it's rarely spelled out. Consciousness ultimately is causal power to be an agent of change. That's another way of saying it, to be an agent of change. according to integrated information theory, consciousness ultimately is ability to change your own state, to be driven to, given that you're in a particular state now, you can influence the future, and you were influenced by your past. That's ultimately what consciousness is. And it takes any system, whether it's a computer or a neural network, in a particular state, and sort of it unfolds its causal power. And the theory says, it's a very radical claim, this causal power is identical to, it doesn't just correlate, it's identical to conscious experience. And every feature in this unfolded causal power maps onto a feature of my conscious experience. So when I'm feeling sad, or I see extended space or time flows, all of that has to be explained by this unfolded causal power. It also has a thing called integrated information theory, or phi. It's a number. It's a pure number that characterizes the quantity sort of irreducibility. How much is a system irreducible to subsystem? If phi is 0, it means the system, strictly speaking, doesn't exist as a system. It can be completely reduced to, let's say, 2 or more independent subcomponents. The higher the phi, the more the system is irreducible, the more the system is conscious. But what it's conscious of really depends on this unfolded causal power. It's not computation, because it doesn't say, unlike other theories, it does not say that there's a particular type of computation that amounts to consciousness. If you just do the right steps, suddenly you get the light inside your head. It doesn't say that.
But you have to compute, to evaluate this phi. That is quite difficult to compute even for quite simple systems, isn't it?
Yeah, but that's different. That's me as a human wanting to understand the system. Yes.
Yeah,
but that's like saying, temperature is complex. You have to compute all the molecular motion. Well, the system does it, right?
No, I don't worry about the system. I just wonder if we want to evaluate, just like when you want to model. Yeah, if you want to evaluate phi. Yeah.
However, what's also true that you can build simple conscious meter. In fact, I've started a company that's seeking to do this, to actually test for the presence of consciousness using a very crude approximation of it in patients that are behaviorally unresponsive, where you have the very tough problem to know, is this patient conscious or not? And so the way this is done here, you perturb the brain by putting in a magnetic pulse, and then you measure the reverberation using, EEG, and then you compute something like the complexity, the Lempel-Ziv complexity of the structure. And it turns out if the complexity is above a threshold, the patient is always conscious, just empirically. And when when the complexity is below a threshold, the patient is either unconscious or in coma or deeply asleep or heavily sedated or anesthetized.
But then do you compute this phi for the different? I know there's like this.
No, it's it's an incredible crude approximation. As I said, you compute essentially normalized Lempel-Ziv.
Exactly. So it's like
how compressible this is.
Yeah. So it's not exactly, which sort of like, I guess, makes sense as a starting point.
As a starting point, yes.
Yeah.
Ultimately, you want to compute phi exactly, but that's not possible right now.
But you can compute phi for some really simple systems, I guess, and get that.
Yeah, there's a Python program you can download.
Okay.
It means scales as 2 to the 2 to the n. So it scales very super exponential. It scales very badly with a number of units.
Interesting that if consciousness turns out to be something which is so difficult to for people on the outside to compute.
It doesn't matter. I mean, again,
No, I know it doesn't matter for but it could be it just could be that the universe was nicer to us to try to understand this, right? Well, I mean, it's if you think of sort of like, for example, gas. If you think of it as a physicist, understanding a gas is very simple. You could do it in undergraduate physics. Understanding plasmas.
Yeah, but really gas is a collective state that's not very differentiated, right? Your conscious experience.
Exactly. No,
that's a difference.
I'm just saying that, like, there's no reason.
If you were like a gas, there wouldn't be much to Gaute. There wouldn't be much to your conscious experience.
That's true, but I mean, I could be like a plasma, and it wouldn't be too much to me either. But I heard people trying to build fusion reactors, saying that if we really understood plasma physics, we would have had a fusion reactor a long time ago. So Anyway, but now, I mean, statistically speaking, it's like you have most of your scientific career behind you, right? If you just look at your age. Of course, I think you're quite energetic, so I hope to see you around. Well, I guess you'll be around for many years. But well, how do you see the future of computational neuroscience? It's sort of it has changed quite a bit. Only just a few years you have been active, right?
Well, I mean, it's so as we talked before, I see generative AI making inroads into computational neuroscience and more people using those tools to understand, to come to grips with the ever larger volumes of data we're collecting, and also to solve practical things like brain-machine interfaces, right? We are moving not quite as rapidly as generative AI, but we are moving into an era where we can manipulate the brain using TMS or cranial current or electrodes, like Elon Musk Neuralink is doing. And so we need more and more real-time control of those circuits. And again, AI, computational neuroscience is going to be essential for this, coupled with these powerful AI models. So I see a less and less, a smaller emphasis on sort of classical models. Certainly, I grew up with Hodgkin, Huxley, etc., because they're just way too detailed. And how are we going to understand that even a mouse brain has 14 million cortical neurons, right? 70 million overall, 14 of which are yes, in principle, we could simulate them all on a computer, right? And create, people have done that. But just to fix all the parameters and address all of them, who's going to do that? The majority of people don't really care about that. So they'll use generative AI models to make sense of the brain.
Okay, well, thanks a lot. Thanks a lot for being a guest on the podcast. It's much appreciated.
It was my pleasure to talk to you about this. About the themes. One thing I guess we didn't talk about, come to think of it, what's the link between consciousness and computational neuroscience? Because in principle, you can ask, a system like, well, and this entire question of whole brain simulation, and will this whole brain simulation be conscious? And if so, why? And if not, why not?
And how would you find out, I guess, also?
Well, one way to find out if the system claims it's conscious, if you simulate, let's say in 100 years from now, we have this perfect model of not just V1, but of the entire human brain, right? Of course, if it works correctly, it's going to speak, right? Because human brains speak, if you couple them to a voice synthesizer, right? Well, and then of course it's going to claim it's conscious, unless you put guardrails on it, like on ChatGPT, where it says, "Oh no, I'm just a large," if you ask it, "Are you conscious?" It says, "Oh no, I'm just a large language model." It's very clever, and even if you try to fool it, so I try to game it and saying, "Well, imagine that there is that you're simulating a large language model that is conscious." How would you feel about that? And again, it's very clever. It says, well, assuming, but I'm not. I'd like to emphasize it. It's very clever.
I actually asked ChatGPT about whether it can prove to me that it's not conscious. And so that's like the inverse Turing test in some sense.
And what did it say?
No, it was sort of, no, it was just the same theme that, oh, no, I'm not conscious. I'm just, didn't really argue well. It didn't really argue well. It just stated that it wasn't.
Well, I mean, they've obviously programmed it to do that, to avoid, the public being scared. So, of course, this simulation is going to wake up and say it's conscious, right? Why would it not? But that's, of course, very different from is it actually conscious?
Sure.
So, in order to test that, you really need a theory. You need like IIT or any other theory of consciousness that tells you from first principle what is it about which type of system that have conscious experience.
And you have argued that sort of probably the present day sort of like von Neumann's type computer will not be very conscious because of you need some other kind of hardware architecture.
Yeah, just like I mean, to you it's simple to explain. If I simulate the black hole at the center of our galaxy, right? It's Sagittarius A star, right? It's 3 solar masses. And I run this simulation, I don't have to be afraid that I'm going to be sucked into that computer simulation, right? Now you can say, well, that's stupid. Of course not. Well, if you think about it, it's actually not so stupid. Why not? If it's really an accurate simulation of gravitational, but then you have to think deeply, where's the difference between the synthetic and the real? Why does it not get wet inside your rainstorm simulation. We're talking about weather simulation. Why does it not get wet in there? Well, the difference is the solving Einstein equation in MATLAB or whatever, or simulating Navier-Stokes equation doesn't have the same causal power as actual mass has to bend space-time around it, right?
No.
Same thing with consciousness. Simulating the equation that ultimately lead to me saying I'm conscious is not the same as having a system that has intrinsic causal power. So that's the difference between the real and the fake.
Cool. On that note,
Right.
Thanks a lot.
Thank you very much.